Hello everyone, welcome to part 2 of dark reaction. In this video, we will be studying the photorespiration. In the previous video, we started the dark reaction and done the Kelvin cycle. If you have not studied the Kelvin cycle and the light reaction, please go back to my channel and watch those videos. Let's proceed forward towards the respiration. If you remember, in the Kelvin cycle, we studied enzyme Rubisco, which was catalyzing the carbon fixation. The first step of the carbon fixation, forming the two PGA molecules. In this reaction, this Rubisco enzyme acts both as the carboxylase and the oxygenase in the presence of oxygen. When the oxygen concentration is high, it acts as an oxidizes. You can see it. RUBP is 5-carbon molecule. It results in the formation of two products, the 2-carbon phosphoglycolate and 3-carbon phosphoglycerate. This photorespiration is also called as the phosphoglycolate or the PCO cycle. It occurs inside the peroxisomes, chloroplast and the mitochondria. The receptor molecule is RUBP. You can see in this slide, in the normal Kelvin cycle, the one molecule of RUBP along with one carbon dioxide. This 5-carbon RUBP is 5-carbon and carbon dioxide is 1 carbon, resulting in the formation of two phosphoglycerate molecules. But in this, only the 5-carbon recover. This one PGA and one phosphoglycolate are formed. Means the 2 carbons are wasted for each molecule of RUBP. But if we take the 2 molecules of RUBP, the 4 carbon atoms will be wasted as the 2 molecules of phosphoglycolate. Out of this, along with this, the 2 molecules of 3-PGA will be formed. So, out of the 4 carbon atoms, the 3 carbon atoms can be recovered through a series of reaction, forming the 1 molecule of the 3-PGA and 2 carbons, and the 2 carbons will be wasted as the carbon dioxide, 2 molecules of carbon dioxide. Let's see that. The photorespiration is a net loss of 2 carbons. If we take the 2 molecules of RUBP, if we take 6 molecules of RUBP, then the 3 carbon atoms will be lost. For 2 molecules, let's proceed forwards. We have already discussed a brief about the Rubisco enzyme. The Rubisco enzyme acts as both as the oxygenase and the carboxylase. When the oxygen concentration is more, instead of carbon dioxide, Rubisco picks up the oxygen molecule and the photofossilization occurs. It resulted into the loss of already fixed carbon dioxide molecules. Here you can see when the carbon dioxide to oxygen ratio is high, means the carbon dioxide content is more. When the temperature is low, the carbon dioxide fixation occurs, the Kelvin cycle occurs, normal Kelvin cycle occurs, and the Rubisco acts as the carboxylase. But in case of the low carbon dioxide to oxygen ratio, when the temperature is high, the Rubisco acts as the oxygenase, catalyzing catalyzing the photofossilization, the photorespiration. Let's proceed forwards. This is the overview of the photorespiration. What occurs here? See, when the oxygen concentration is high at the high temperatures, the Rubisco will act as a oxygenase, and if we'll take the two molecules of RUBP, the 5 carbons will be it is a 5 carbon molecule, so 5 into 2, 10 carbons are there. We have a net of 10 carbons. The 2 PGA molecules will be formed, means 6 carbons are recovered. The 2 molecules of phosphoglycolate will be formed, each having the 2 carbon atoms. Thus, the 4 carbon atoms are wasted. Out of this, the 3 carbon atoms are recovered through a series of reactions. And still the one molecule of carbon dioxide is wasted. Here, the phosphoglycolate converts into the glycolate, which enters the peroxisome from the chloroplast. The reaction of the photorespiration initiates in the chloroplast. Then the glycolate enters the peroxisome, where it converts to the glyoxylate. Then it undergoes the amination, forming glycine. The glycine enters the mitochondrion. It converts into serine. The serine enters the peroxisome. The serine gets converted into hydroxypyruvate, which converts into glycerate. Glycerate enters the chloroplast, which converts into the phosphoglyceric acid. We have seen the products of this reaction. Let's see what reactions occurs during the formation of these products. First of all, the phosphoglycolate undergoes the dephosphorylation, forming the glycolate. The glycolate enters the peroxisome. The glycolate undergoes the dehydrogenation, releasing the peroxide H2O2, forming the glyoxylate, that is also the 2 carbon molecule. It resulted into the formation of the glycine by undergoing the amination or the transamination. It enters the mitochondrion, from where the glycine, the 2 carbon molecule, we are having 2 moles of glycine, which are having 2 carbon atoms. The 2 molecules of glycine forms the 1 molecule of serine, and the 1 carbon atom is released as a carbon dioxide as a result of decarboxylation. Here, 3 reactions occur: deamination, dehydrogenation, and decarboxylation. Now the serine enters the peroxisome, where it undergoes the deamination, forming the hydroxypyruvate. The hydroxypyruvate converts into a glycerate. Glycerate enters the chloroplast. In the chloroplast, the glycerate undergoes the phosphorylation in the presence of ATP, forming the phosphoglyceric acid. Thus, out of the overall 10 carbon atoms, 9 carbon atoms are recovered. The 1 will be wasted as carbon dioxide. Let's take a brief of photorespiration. It is a wasteful process, as it resulted into loss of carbon atom, already fixed carbon atoms. So let's begin with this. Photorespiration initiates in the chloroplast. The Rubisco attaches to the RUBP, acts as the oxygenase. Oxidation reaction occurs. The 2 molecules are formed. The 3 carbon atom 3-PGA and the 2 carbon atom phosphoglycolate. The 3-PGA is a normal intermediate of Kelvin cycle, but the phosphoglycolate cannot enter the cycle, so they are removed. To recover the lost carbon atoms, the phosphoglycolate undergoes a series of reactions under various organelles such as the chloroplast, peroxisome, and the mitochondria. The 3/4 amount of the carbon atoms are recovered. Out of the 4 carbon atoms, 3 are recovered. One is lost as a carbon dioxide molecule. By this, we have already discussed the photorespiration. In the next video, we will be studying the C4 cycle. That's all for today. Please like, share and subscribe to this video, and please subscribe to my channel. Please mention in the comments if you want any specific topic to be discovered. That's all for today. Thank you.